Technical Veterinary Learning. Founder and producer is Prof.Ahmed Mamdouh El-Sherif, Professor of Epidemiology and Infectious Diseases, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University. We will explain, discuss, and give complete knowledge about Salmonella abortion. Salmonella abortion, it is an infectious disease of sheep characterized by abortion. Epidemiology of Salmonella abortion. Number 1, pathogenic risk factor. Salmonella abortion caused by Salmonella microorganism, which is gram-negative, rod-shaped aerobic bacterium of family Enterobacteriaceae. Salmonella microorganism belonging to family Enterobacteriaceae. Salmonella abortus ovis or Salmonella enterica serovar abortus ovis. This is number 1. Number 2, Salmonella typhimurium. Number 3, Salmonella Dublin. Number 4, Salmonella Montevideo. These 4 species are the main pathogenic risk factor for Salmonella abortion in sheep. This is the shape of Salmonella microorganism under the 3D microscope. Salmonella species microorganism under 3D microscope moving in bloodstream. Number 2, occurrence of the disease. Salmonella abortus ovis infection has a worldwide occurrence and has a generally low prevalence. Appears to be more common in some European and Western Asian countries. Susceptibility of Salmonella abortion disease. The pathogen is highly adapted to sheep as a host specific. Transmission of Salmonella abortion. In case of sheep, infected animals that are introduced newly to the flocks naive to the pathogen. Reservoir of infection. Number 1, infected animals that are not aborted. Number 2, Salmonella organisms persist in internal organs of symptomatic carriers for up to 6 months. Number 3, Salmonella organisms excreted in the feces and vaginal mucus for periods up to 4 months. Number 4, the predisposing factors for Salmonella abortion disease. Number 1, different stress factors. Number 2, introduction to infected animal to naive flocks. Number 3, contaminated water supplements. Sources of infection. Salmonella abortion spreading routes are: oral route is the main route. Number 2, conjunctival. Number 3, respiratory. Number 4, contaminated food and water staffs. Number 5, venereal. Diagnosis of Salmonella abortion. Number 1, clinical symptoms. Clinical symptoms of Salmonella abortus ovis infection. Number 1, abortion during the second half to last third of gestation period. Number 2, lambs may be also still born or dying within the first day of life. Number 3, mortality in lambs is common from either weakness, ensuing hypothermia and or hypoglycemia, or due to development of acute pneumonia in previously healthy lambs up to 2 weeks old. Clinical signs of Salmonella abortus ovis infection. Number 4, influx. Where the pathogen is endemic, abortion occurs sporadically, mainly affecting primer, preus, and newly introduced ewes. Number 5, the infection appears to induce a strong immune response, preventing abortion during the following pregnancies. Number 6, fever and diarrhea in case of Salmonella Dublin. Clinical symptoms of Salmonella abortus ovis and Salmonella Dublin infection. Influx naive to the infection, introduction of the pathogen can cause abortion storms, with up to 60% of ewes aborting generally in the last trimester of gestation period. Ewes rarely develop clinical signs, although some may transiently have a fever or develop post-abortive endometritis with vaginal discharge. Septic metritis and peritonitis in dams have been associated with deaths among ewes. Infection with Salmonella montevideum. Number 1, abortion reported at 14 to 17 weeks of gestation period. Number 2, this means that infection occurs at 12 to 14 weeks before. Number 3, abortion is the only sign of this disease. Infection with Salmonella typhimurium. Number 1, dams may be found symptomless death. Number 2, systemic reactions may be noted, such as fever, diarrhea, apart from the flock of food, and etc. Number 3, abortion is the mainly signs. This figure showing aborted lamb due to Salmonella infection. This showing aborted lamb due to Salmonella infection. This figure showing aborted lambs due to Salmonella infection. We can see on the ground aborted lambs due to Salmonella infection. This movable picture also showing aborted fetus due to Salmonella abortus infection in sheep. Laboratory diagnosis. Corrected samples gathered for lab diagnosis of Campylobacteriosis are blood, serum, uterine discharges, aborted materials, gallbladder, mesenteric lymph nodes. There are 6 laboratory tests applied for diagnosis of Campylobacteriosis. Number 1, microscopical examination of Gram's stained smears. Number 2, isolation by culturing on gram-negative media. Number 3, agglutination test. Number 4, complement fixation test. Number 5, ELISA test. Number 6, PCR test. This picture showing Salmonella stained by Gram's stain under the microscope, where gram-negative bacilli are very clear here in this slide. Salmonella species grown in blood agar plates. Using of PCR to identify Salmonella abortus ovis is visible because the organism has an 200 elements as a distinct chromosomal location. The resulting of PCR assay has high specificity for Salmonella abortus ovis, effectively infectious diseases primarily affecting the reproductive system, discriminating it from other Salmonella enterica serovars. Identification of Salmonella by phenotypic differentiation and PCR. Multiplication of Salmonella target DNA by polymerase chain reaction with primers that amplify a chromosomal region which contains the Salmonella abortus ovis specific IS200 insertion the lane from 1 to 10 examined isolates of Salmonella abortus ovis, lane 11 Salmonella enteritidis, lane 12 Salmonella abortus ovis ZG subspecies 355, NK it is a negative control 200, 600 and 900 base pair. Phylogenetic tree of Salmonella species as appear here. We see Salmonella Montevideo and Paratyphi, Salmonella Newport and other Salmonella species. Post-mortem examination for Salmonella abortion disease. Number 1, there are no distinguishing lesions by naked eye either in the aborted fetus or the placenta. Number 2, distended gallbladder can be detected by the sonar before death. Number 3, swollen and friable liver. Number 4, necrotic foci may be noted in the liver in case of Salmonella abortus infection. Number 5, inflamed mesenteric lymph nodes and intestine. Distended gallbladder as seen by sonar examination before death or slaughtering. Aborted fetus due to Salmonella infection. Differential diagnosis of Salmonella abortion disease. Salmonella abortion have to be differentiated from 7 diseases that cause abortion in the sheep. Number 1, listeriosis. Number 2, campylobacteriosis. Number 3, toxoplasmosis. Number 4, leptospirosis. Number 5, ovine brucellosis. Number 6, fungal organisms as aspergillus and other mycotoxins. Number 7, Q fever disease. Treatment of Salmonella abortion. Treatment of Salmonella abortion have to be done through the following 3 main steps. Number 1, the administration of broad-spectrum antibiotics might aid in controlling of an outbreaks, but available reports are not generally encouraging for that. Number 2, chloramphenicol and the trimethoprim as well as sulfadiazine combination with non-steroidal anti-inflammatory drug are considered effective for treatment. Number 3, use of chloramphenicol in animals intended for human food production is not permitted in many countries. Antimicrobial agents used against Salmonella abortus ovis are number 1, ampicillin, number 2, amoxicillin with clavulanic acid, number 3, cefotaxime, number 4, colistin, number 5, oxytetracycline, number 6, streptomycin, number 7, neomycin, number 8, nalidixic acid, number 9, enrofloxacin, number 10, spectinomycin, number 11, sulfamethoxazole with trimethoprim. Control of Salmonella abortion. Number 1, application of complete hygienic measures such as during an outbreak aborted ewes should be isolated and abortion products that contain large amounts of bacteria must be destroyed. Number 2, disinfection of stools and vomits with an agent with proven efficacy against Salmonella species is very important. Number 3, a live Salmonella typhimurium vaccine with optimal level of attention for sheep constructed by means of metabolic drift mutation was highly effective in preventing Salmonella abortus ovis induced abortions under field trial conditions. Number 4, subcutaneous and conjunctival vaccination with a live attenuated strain of Salmonella abortus ovis confers immunity for at least 3 lambing periods. Number 5, more recent vaccines, including those containing plasmid cured strains of Salmonella abortus ovis, are effective in preventing pregnancy losses in response to experimental challenges with wild type of Salmonella abortus ovis. Number 6, the results of vaccination need to be very carefully appraised because flock immunity develops rapidly and the disease tends to subside naturally in the second year. Number 7, avoid contamination of food and water. Number 8, control of rodents. Number 9, elimination of carriers. This is the shape of Enmiva vaccine, which is inactivated vaccine against Chlamydia abortus and Salmonella abortus ovis in sheep, in suspension for injection at last third of the gestation period. The administration route is subcutaneous injection. The dose is 2 ml per animal. Withdrawal period in meat is 0 day. Contraindications: don't use in case of hypersensitivity to the active substances and or any excipient. This is the shape of Ovivac vaccine. This is inactivated vaccine to prevent abortions caused by chlamydophilia and salmonella in injectable suspension. The dose in case of cattle is 5 ml per animal as of 6 months of age. In case of sheep and goat is 2 ml per animal as for 6 months of age. Keep on the track by following us in our technical veterinary learning channel to get the most newest knowledge about any animal infectious diseases throughout the world.